extent, but there is a small variation that you will see in future. You see this curly wing and with bristles. So this and this is sepia eye color with curled wing. So curly wing and curled wing are two different varieties of mutations. So here you see that there are vestigial wings also, but sepia color eye also. So double mutant. This is called as double mutant. Here also you have sepia colored eye and curled wings. So this is also a double mutant. This is, see, this is gene CU and this is gene CY. So there are two different genes. This is curled wing and this is curly wing. So this is curly wing, and then there are bristles also. You can see here bristles, right? So the bristles mutation is also there. Then this is yellow body and white eye and forked bristle. The bristles, if you see under the microscope, they will be forked, right? So this is triple mutant. Then there can be multiple mutants also. So rhaphoid eye, curled wing, claret color, stripe, ebony, all together. So you can have multiple mutations as well. Now, you can, to get aware of how Drosophila functions, can perform several experiments in your laboratory only. I could see that there was one student in the chat box who is probably working on Drosophila. So you can start your work with the rearing and handling of the Drosophila. You can bring Drosophila into lab, make culture medium, start growing it, get a feel of it, observe it under the microscope, let them breed, observe the eggs, then let the larvae come out, then pick up first instar, second instar, third instar larva, observe those larvae under the microscope. Then if you have come across some mutants, you have some mutants in your laboratory, or if you can order some mutants, then go and look into some mutants, their phenotypes also, try to observe them. Then you can solve some problems also, and not only solve some problems, but also allow some mutants to crossbreed together and see if there is some Mendelian inheritance taking place or not, or whether linkage is taking place or not, whether 2-point linkage is taking place or not, right? So there are several possibilities with respect to the genetic crosses as well. And based on that, you can analyze the recombinant frequencies from a trihybrid cross and calculate the genetic distances as well. Then heat stress tolerance in Drosophila. As I said, the Drosophila does well at a temperature at, say, around 25 degrees Celsius. So what happens if you increase the temperature, say, to example, 30 degree, then to 35 degree, then to 40 degree? Do they faint? Then if they faint, how much time do they require to revive again? Similarly, if you go bring it down to, say, 15 degrees and 10 degrees, and correlate this with the time duration for which the temperature has been increased or decreased, you will see that there are different responses. So there are temperature responses as well. And like I was telling you, the olfactory responses, the larvae are extraordinary in identifying some chemicals and moving only towards. So you can perform plate assays, where on the plate you will have agar along with, say, some yeast, and then release the larvae at the center, and you can have say, attractants on one side and repellent on one side, and you will see that larvae easily move towards the attractant, some of the chemicals which Drosophila prefers. Then there is another study which we call as negative climbing assay. Climbing assay is where, like I said, Drosophila, no, though they prefer to move upward, right? If you put them in vial, they will always be going upward. So if you bang them on the floor, they will start climbing up. But if you keep banging them, probably they will after some time lose their ability to climb up. It will take some time for them, right? So they are negatively geotaxic. Negatively geotaxic means they will move away from the floor, away from the gravity. But then if you give them some harsh treatment, probably they'll take some time to get up and move again away from the ground. So you can perform similar such studies. Like I said, Galfor system has been established wonderfully well in Drosophila melanogaster. So if you can introduce GAL4 gene into Drosophila and induce ectopic expression, I don't have image right now, but then you can have a Drosophila with eye on a leg, maybe, right? So their eyes get expressed on the leg. This is called as ectopic. Ectopic means something which is not the normal place, which we refer sometime to the pregnancy also. That means normal pregnancy takes place in uterus of human body, right? So if the pregnancy takes place in fallopian tube or some other organ, you call it as ectopic pregnancy. Similarly, in Drosophila, you can express eyes in some other part, right? So that is also another interesting part. And you can perform these interesting things in laboratory as well, where you can take transparent nail polish and put it on the eye of the Drosophila and slowly peel it off, so that you will see all omatidia, and you can easily count the omatidia also, how many hexagonal structures are there, small units are there, right? These are the two experiments that we perform every now and then, the mitotic chromosomes and the polytene chromosomes from Drosophila. Now the established protocols are there to extract this genomic DNA from a single fly. You don't need to have a lot of flies and put them in liquid nitrogen, crush them, and you can also take out DNA from a single fly as well. Rapid extraction methods are there. And similarly, you have several other interesting experiments. For example, pH profile. You require simple stains which can be used to track the pH sensitive stains are there. So you simply just use those stains and they will stain the intestine with different colors, so that you come to know that which part of the intestine is acidic, which part of the intestine is less acidic or towards the basic side. For example, intestine later part, it will be basic, and the earlier part would be comparatively acidic, and so on. So these are some of the interesting experiments that one can have in Drosophila. And that brings me to the end of today's presentation. And I hope I could give you some information on model organisms. I've shared some details of at least 4 to 5 model organisms today, and more emphasis on Drosophila, because recently I have been fascinated by Drosophila. Earlier I worked on fishes, I worked on microbiome, I worked on bacteria, then now switching over to Drosophila. So I thought I will share some information with you on Drosophila as well. So I